Good afternoon. It's a great pleasure to speak at this workshop where we have such an incredible mix of people coming from all kinds of directions. So, ranging from very, very general abstract theory, almost bordering on philosophy, to great experimental sciences. We have wet biology, as we have heard in David's talk just a few moments ago. And before that, we have seen very abstract, but I think very important theoretical framework from Steve Frank. So what I'm going to discuss today is somewhere, in my view, right in the middle. It's primarily and mostly analysis of empirical comparative genomic data, but with some generalizations, some excursions into theory and even into philosophy, as you will see. So basically, I'm going to describe three generalizations of what I consider among the major models and major concepts of molecular evolution. Namely, the very concept of the genome and how we generalize on that. The concept of the tree of life and how this changes. And the concept of molecular clock and how this also may be replaceable by something more general. And my main point, basically, is to try and convince you that the conceptual shifts in evolutionary biology that I think are in the making are very much data-driven. Genomics drives evolutionary biology away from the useful but old, rigid constraints and models that are becoming outdated to models that are effectively based on statistical properties of ensembles of genes and genomes. So, I'll probably have included here too much stuff to actually discuss during this talk. I think everyone wants to have the reception, so it would be inhumane. And I'll mostly concentrate on this first part, on what I consider to be a paradigm shift, at least in microbial evolution, from genomes to pan-genomes to what I call the gene universe. And there's quite a bit of new stuff that I really want to share, and the rest is largely published business. So I'll go through that very briefly. So anyhow, in the 20th century, the way people talked about evolution, the way people described evolution, was primarily based on the evolutionary history, phylogeny of individual genes, such as above all 16 sRNA, a universal highly conserved molecule that was considered to be the best model for reconstruction of the history of life, or the tree of life, if you will. Well, in the 1990s, thanks to the new technologies, we have become aware of the genomes and of the different evolutionary histories of various parts of the genomes. Finally, these days, in the second decade of the third millennium, we have to replace, I think, the notion of the genome with the notion of the pan-genome. It will become clearer what this means during the rest of this talk. But basically, the pan-genome is the entirety of the genes that is available to a particular group of organisms. Primarily, this is used when we speak of microbes, but actually it's a very general concept. The entirety of the genes that is available to, let's say, to be more concrete, to a species. So, in other words, basically, the concept of the microbial pan-genome is well described in the abstract of this nice paper. A bacterial species can be described by its pan-genome, which is composed of a core genome containing genes present in all strains and a quote-unquote dispensable genome containing genes present in 2 or more strains and genes unique to single strains. Given that the number of unique genes is vast, the pan-genome of a bacterial species might be orders of magnitude larger than any single genome. So that's the idea. Where is the reality? The reality is that pan-genomes of different microbial species are different. And this is illustrated here by the pan-genomes of 2 well-characterized bacterial species, Bacillus anthracis and Streptococcus agalacticae. And you can see that as strains, this is the number of strain genomes sequenced, and this is the size of the pan-genomes, the number of genomes discovered, so to speak, by these genomes. You can see that they're drastically different. These pan-genomes. While in Streptococcus, when new genomes are being genomes from new strains are being sequenced, the number of discovered genes, the size of the pan-genome, grows and grows. In this case, at least, without visible signs of saturation. Usually, this is called an open pan-genome. If you try to extrapolate from this, it's sort of infinite, although, of course, nothing is. The Earth is finite itself, so nothing can be literally infinite, but we don't see the signs of saturation for these open pan-genomes. This is dramatically different. After sequencing a few strains, you basically don't discover anything new. So this is a closed pan-genome. In practice, in real genomic sequencing, the considerable majority of the pan-genomes seem to be more like this than like this, more open than closed. We shall, towards the end of this part, we shall come to the estimates that may show you how large, actually, how vast these seemingly open pan-genomes actually might be.
Question?
Please.
Can the notion of pan-genomes be defined for mammals?
Yes, of course.
And does it grow as the first one or the second?
I would have to just get it done. Certainly, you don't have to go far. David already presented in his talk evidence that there are pan-genomes that are bigger than any individual genomes by showing the birth of a new gene. And we have heard about birth of new genes previously, even during this workshop. My intuition is that it is not quite like this. It's more in between, but sort of closed. But I haven't done the analysis. Okay, so from the pan-genome of a species, we actually can go further and look, in a certain sense, at the structure of the gene universe. And by gene or genomic universe, by genomic universe, I mean the entirety of all genomes on the planet. And of course, we can try to look at some kind at different depths in this genomic universe. We can try to look at small neighborhoods, large neighborhoods, and some representation of the entire universe. So if we try to do so at different levels and make plots like this, where here the horizontal axis is the number of organisms in which a gene is represented, or to be precise, by gene I mean a cluster of orthologous genes, which may be called the same gene in all genomes that have it. So the number of genomes in which a gene is represented, and the number of genes that are represented in exactly this number of genomes. So when we do this gene frequency plot for a representative set of the entire prokaryotic part of the genomic universe, we detect this tripartite structure. So Steve in his talk showed us a variety of beautiful and not so beautiful distributions. Those were, however, all simple, all representing apparently a single attractor. Here we have something more complicated. It roughly can be described by three exponential functions that correspond to the tiny, highly conserved core of nearly universal genes by a much larger part that I fancifully call the shell of these genomes, about 2 orders of magnitude bigger, and by something that I call a cloud, which is even much bigger. Here I included only the genes that are represented in at least 2 genomes. If you include the orphans, those that are found in a single genome, the cloud will be really big in this case. The more interesting part of this, and that...
Can I start, Eugene? Is this reasonably insensitive to where you put the cutoff of defining genes as being in the same category, or is that...
Yes, this is very insensitive. This is actually highly robust to this. Well, and I'm not just hand waving. We have done it in at very different levels of crudeness. You'll see the same kind of thing. In addition to being highly robust to that, and less trivially, this structure recapitulates at any level of, at any depth at which you would like to peer into the genomic universe. You can peer as far as you can limit yourself to one domain of that universe, let's say Archaea, and the slopes of these exponents, the sizes of these parts will be different, but the tripartite structure, sort of a symmetrical U-shaped distribution, will come up inevitably whatever you do. And this is practically a pan-genome, according to the previous definitions. Pretty much. This is pretty much at the level of a single species. And you'll see the same kind of tripartite structure. And you'll see more of this. So I like to speak of a fractal structure of the prokaryotic gene universe, because this structure repeats itself at all different levels. Incidentally, I think this was first explicitly published in this book that also contains a number of other ideas on evolution. All right. Just to give you an additional perspective on the diversity of the structure and diversity of the gene universe. Here I put together this gene frequency plot for the cellular part of this universe and for the viral part. As you can see, they're quite different. Here you do not effectively have this tripartite organization. This part is severely, pretty much completely suppressed. This part is greatly enhanced, indicating that the world of viral genes is even much vaster than that of the genes of cellular organisms. But this fascinating part of the gene universe is for a different talk, and in what follows, I will discuss that other part, and first ask this question: where does the universal fractal architecture of the gene universe with skewed U-shaped distribution of gene frequencies come from? So in order to get some handle on the answer, we developed something that I call the SGT, Stationary Genome on a Tree, and that's a computational model that limits the changes of genome size to some small margin that is that is realistic in the case, reasonably realistic in the case of microbial evolution, and simulates evolution along a defined evolutionary tree by taking genes from an infinite reservoir and replacing the genes in your evolving genomes under different models. And the simplest one is the neutral model, neutral in the sense that all genes have the same replacement rate. And then there are models of selection with different levels of complexity. Selection in this case means that genes differ in their value, so to speak, for the evolving genomes, that is, in their replacement rates. We can take a gamma distribution of replacement rates, we can take a 2+ model, where there are important genes and unimportant genes, so to speak, and we can take something a little bit less unrealistic, where replacement rates are taken from a stochastic population model, and this, the ratio of the population size to genome size, becomes the measure of selection strength. And then we do apply this to real data from a number of microbial groups, and here there is where these different cutoffs come into play, because this was done with a somewhat different approach than the distributions I showed previously, and this does not really change, at least in terms of the general shape. So when we fit the predictions of these, or the results of the model evolution under each of these models to the empirically observed distribution, you can see immediately that the neutral model with these red symbols do a really bad job by not properly modeling the highly conserved classes at all. Well, in order to compare different models with selection, we have to apply something slightly more sophisticated, in this case the Akai key information criteria or another information criteria, and what I plot here is the difference, is the advantage of these models over the neutral model. In this case, for each of these models, the advantage is absolutely overwhelming. It translates to the probability of this happening by chance, to pathological probability of this happening by chance, 10 to the minus 200 or something. But we also can see that the more complex selection model wins with statistically significant margin over the very simple toy implementations of selection. So,
sorry, can you go back to the previous slide? Because I don't understand what we're supposed to be looking at, the previous one. I don't understand what we're supposed to be looking at here.
Oh,
as far as which is data and which is...
Oh, yeah. Sure.
Sure.
Yeah. The histograms are empirical data. These black columns.
But class K is meaning something you didn't introduce that before.
Yeah, it does not matter. This simply the number of genomes. What we did was taking groups of 10 genomes and plotted these frequency distributions.
You gained the continuous distribution from the previous slide.
Yeah, that's correct. These genes, these are absolutely, in terms of this group, absolutely conserved genes. Those that we find in 10 genomes, these are the least conserved, those that we find in 1 genome, and everything in between.
And for organisms where you know the phylogeny?
Yes,
Okay.
absolutely.
I think you know the phylogeny.
Absolutely. We have some confident phylogeny for these organisms. Okay, so the conclusions from this is that unlike some other universal distributions of genomic variables, again harking back to Steve's talk today, this tripartite distribution is substantially affected by selection. In a sense, if using the same language, I dare say that this is not aggregation of random, noisy random variables alone. There is substantial modulation over this that we can detect. All right. And now I want to describe, in a certain sense, what we described there was the statics of this genomic universe. What I now want to discuss is some new data that I think give us some insight into the genome dynamics and allow super-genome size estimation. So in order to do this analysis, what we used is the so-called ATGC database, which we developed a few years ago and continue updating. I think this is a useful thing. ATGC doesn't stand for what you think it stands. It rather stands for alignable tight genomic clusters. And these are clusters of related, fairly closely related prokaryotic genomes that not only didn't evolve from each other very far in terms of sequence, that are specifically not saturated in terms of KS, of synonymous positions, but that also maintain a substantial degree of gene synteny. Very similar related genes. For instance, just to be specific, all the E. coli strains and Shigella strains will be in one ATGC, but Salmonella, to which we compared on one of the preceding slides, is already part of a different ATGC. What we do then is the evolutionary reconstruction from the comparison of these genomes. Basically, this is not so critical. We do some comparisons with this, but these two components are essential. We make a species tree, in this case by using a concatenated alignment of conserved genes for each ATGC, and then we make this matrix of gene presence absence. In this case, I don't say gene, but I say COG, cluster of orthologous genes, so one side of this matrix are species or strains that belong to a given ATGC. The other side are the clusters of orthologous genes, or you may say simply genes, and you put 0 here if the corresponding gene is not represented in the given genome, or you put 1 if it's represented with 1 paralog, or 2 if it's represented by 2 paralogs, and so on and so forth. Then this allows for evolutionary reconstruction that primitively can be done by maximum parsimony, and a bit less primitively can be done by maximum likelihood of certain genomic by obtaining likelihood of certain of the rates, maximum likelihood estimates of the rates of certain genomic events, such as gene gain, duplication, and loss. I will speak about this in slightly greater detail in a moment. Specifically, they use the software code, maximum likelihood software code count, developed by our collaborator Miklos Chouros from the University of Montreal. This maximum likelihood model, maximum likelihood reconstruction, is based on this very simple model that, however, I think is very important for understanding the evolution of genomes and gene families, the birth and death, or sometimes we say birth, death, and innovation model of gene family evolution. It's extremely simple. You basically have these 3 rates here, the death rate, the birth rate of a gene family and the expansion and reduction rate. In this case, the gene death rate is the same, is set to be the same for families of size, so it's the same parameter, and genes, new families come from the super genome, from this bucket that is the super genome. So here I try to distinguish between the two terms. Pan-genome, which is the totality of the discovered genome, discovered genes, namely that is an observable quantity, and the super genome, which is a sort of a hypothetical quantity, the entire reservoir of available genes, the limit to which the pan-genome tends.
So kappa here is the rate of gaining this particular gene family, not a random one, just that one right there.
Yes, indeed. So as we, it will be important. It's not really important for the model, but it will be important for conclusions to realize that family gain effectively occurs biologically, corresponds to horizontal gene transfer, whereas this denotes the process of gene duplication. So you'll see the relationship between these processes. So we derive these maximum likelihood estimates of the rates of all of these processes, and on this slide I plot this against the branch length, or in other words, the number of substitutions per site. And so this measures the ticking of the genomic clock. And we see that there is a strong positive correlation here between the number of gains, number of losses and expansions and reductions, and the branch length. So there is, this correlation is weak, these are strong, but in each case there is a significant clock-like behavior of each of these processes, suggestive of an important neutral component in these evolutionary processes. But there is something else to which I want to bring your attention. And this is the number of events, gene family gains or gene family losses per nucleotide substitution. So you see that then the evolutionary distance is, let's say, 1/20th of a substitution per nucleotide, a very short evolutionary distance. You already have, per that short evolutionary distance, hundreds of these different events, hundreds or dozens of these different events. So, what we have already seen is that the genome dynamics is rapid. 1, 2, there is a clock-like behavior. Another very striking observation that I still cannot fully come to terms with, but it appears completely robust and observable in many of these groups, is the extreme variability of genome dynamic rates. So to speak, lifestyles of microbes may vary from extremely squalid and boring to highly turbulent. These rates differ by several orders of magnitude. And so do the ratios of these different events. But here,
like what organism to what organism, or like what family to what family?
I cannot say. We have in this collection, in what has been analyzed, we have 34 distinct clusters of bacteria, and one archaea. What's available? We analyze everything that is currently available in terms of the sufficient number of closely related strains, and they cover much of bacterial diversity. I cannot say that I see much rhyme or reason in the biological sense in these strongly varying rates. That is, we cannot say that, let's say, proteobacteria are highly dynamic, whereas gram-positive bacteria are very static. Or even parasites are highly static, whereas free-living organisms are highly dynamic. Not so. There are differences, but they're fairly subtle. These huge variations are not explained in this straightforward manner.
How do you know that a lot of the variations aren't from variations in the mutation rate rather than variations in the genome turnover rates?
I am not so sure, Daniel. What do you mean now by mutation rates?
So you when you say of a clock, so that your clock is the number of rate of synonymous substitutions, right?
Oh, well, yes. Yes. One part is what we measure are these events against synonymous substitution.
Right. So synonymous substitution rates can vary by a factor of 100. So I don't believe this hypothesis, but one hypothesis is that the turnover of the genomes is similar, but the mutation rates are very different.
Well, yes, this is relative to the this is relative to the substitution rate indeed, but you have to keep in mind that these are short evolutionary distances. These cover relatively short evolutionary times. So we did not measure here, we did not try to measure the variation of the actual or the sequence evolution clock here. We just took it as a standard. I do not think, however, that along such short branches, DS would vary by 2 orders of magnitude. I would very much suspect
because
that
all of the phylogenies are analyzed under the assumption that it doesn't vary. Okay, but this is a technical question.
This is a very technical question, but again, these are very short evolutionary distances. Of course, the genomic the sequence evolution clock is not precise here, but we actually know that dn over ds is quite a stable feature within these small groups. So I think it holds.
Question?
Yes.
Do you think the statistics can be highly skewed toward genomes that can be cultivated, and non-cultivatable genome may have more turbulent life because they have to live together with other species, react to antibiotics?
No, I do not. There is always this issue, but I do not think so, because here we have a huge representation of lifestyle, parasites and free living. The parasites that live in relative isolation, things that live in our guts, in those microbiomes, things that live in the sea, things that, like archaea, that are not parasites but live in isolation in Yellowstone pools and so on and so forth. So far, whenever someone managed to cultivate something that was not cultivable before, this organism is interesting, but it is not qualitatively different from the cultivable ones. So no, I don't think this is a huge issue. Okay.
How much could this be sensitive to sampling biases in different types? So, for example, around the E. coli are like usually oversampled, and if they just in their ecology they end up picking up or dropping genes, and they're oversampled, it would skew your estimate as opposed to somewhere else where you have a single representative.
Of course, we cannot do anything with single representative or 2 representatives. We start from, I believe, 10. And you're right that there are very many more equals. But I can just tell you that this does not depend on the size of the group. It simply does not correlate. All right. I think I have to proceed faster, because I want to tell you quite a few more things. and not to keep it from the reception for too long. So in any case, there is this huge, still to a large extent unexplained variability. But then there are also these relationships that while also, namely the ratios between loss and gain, reduction and expansion, gain and expansion, and so on and so forth. And here we see two, and these vary, but they vary less. And when we look at the modes of these distributions, or medians of these distributions, or whatever, we actually start seeing striking things. Namely, that the ratio of loss to gain typically is greater than 1. The genomes lose more genes, or more families, than they gain. This is already interesting. What is, I think, especially dramatic and striking is that there is an order of magnitude, notice, order of magnitude more gains of families than expansions. Remember, horizontal gene transfers versus duplication. There is order of magnitude more of horizontal transfers versus duplication. And there is also an order of magnitude more of loss of gene families than there are reductions. So this puts a very specific qualitative aspect on these dynamics. It's very much about loss and acquisition of new families. As already touched upon, we can look along the diversity of the microbes and see considerable conservation of these trends, with variability, of course, especially in terms of the rate of loss. Actually, what we tried to do is a very crude estimate, of course, and to a large extent, as a matter of sort of fun, was to date these events, linking genome dynamics to real time, using the estimates coming from Yersinia pestis, from dated Yersinia pestis evolution, from comparison of the causative agent of the Justinian plague in the 6th century to modern isolates. So these are crude estimates, but I think they give us an idea, and that idea is striking. So we have, so these are values per 1,000 years. More than 10 events of these kinds per 1,000 years. In other words, in the entire microbial world, a huge number of genes are lost and gained on a time scale commensurate with our lifetime. Of course, anecdotally, we already know about that from antibiotic resistance and such things. Interestingly enough, these rates show significant decline with phylogenetic depth, indicating that many gains, losses, expansions, and reductions actually are transient, are not really fixed in the population. Now, we also notice that gene family gain and expansion rates do positively correlate with the pan-genome size. Not the size of the super-genome, so far we don't really know about that, but the size of the actually discovered pan-genome. So that at least rhymes with reason. So we have greater rates of gain corresponding to larger pan-genomes. What we also see is a general genome flux trend. In other words, the rates of all these events, including rates of gains and losses, are positively and significantly correlated. You could think that there are primarily gainers and losers among evolving genomes. And while such things certainly exist, some are gainers, some are losers, that seemingly is not the main distinction. The main distinction seems to be between dynamic and static ones. So we do the PCA in the genomic event space, and it's fairly striking, because the first principal component, in this case, explains over 70% of the variation, which is something that you rarely see, that you, in my experience, which is not inconsiderable, you rarely see in the in comparative genomics. And this first component seems to correspond to the general genomic flux. All these original variables contribute in the same direction and significantly to this PC1. So this PC1 represents visibly what I already pointed out, that the main trend in genomic evolution is the level, the magnitude of this overall flux. The second principal component that is also significant is the balance, indeed, the relative contributions of gain and expansion, loss and reduction. So that's the difference between gainers and losers. And the final thing in this story is the promised super-genome size estimation. You could, this is a very simple approach, and there are different ways to do so, of different levels of complexity, and we're working on that. This is a very simple approach of which you could already guess, I think. Yeah, we do that from the incidence of multiple gains. So if the simplest approach is such that every time a family is gained, drawn a trend from a well-mixed super-genome, I will now use the term super-genome for that reservoir, and then you can easily estimate, easily obtain the maximum likelihood estimate of S. We can use a slightly more sophisticated approach when the gain probability varies between different families and use different distributions for that. I wouldn't go into the details of this. The difference is not huge, but the results are consistent and interesting. This is under uniform gain probability distribution. This is under power law gain probability distribution. For the substantial majority of these groups, there's sort of a new constant emerging, if you will. The size of the super-genome is roughly an order of magnitude greater than the size of the typical genome. Granted, there are several groups for which, as far as we can see, the super-genome remains open. It's quite huge. But the typical trend in the microbial world seems to be this.
So the super-genome is all the ones in that group?
Not
all bacterial genes?
The super-genome is a bit of an abstract notion. It is the entirety of the genes to which, let's say, E. coli or all strains of E. coli have access in the course of their evolution.
It's bigger than the pan-genome, right? It's everything that could get.
Then why isn't it all bacterial? I mean, why isn't it all bacterial genes? Or
because to give you a trivial example,
we haven't seen them coming.
to give you a trivial illustration of that, I do not really believe that over the course of its evolution, E. coli ever had the access, any kind of direct access to hyperthermophilic habitats and the genes that are typical for those habitats. So it's not the entire genomic universe. Or to be more precise, of course, you may say that this is the entire genomic universe, and there is some probability density function over all genes, but in practice, its values are indistinguishable from zero for the great majority of them. That's the statement in more realistic terms.
Since the model has a fixed, I mean, there's some weighted measure over this, which is in practice what you're estimating.
Yeah, right. In practice, yeah, you may say that they're estimating the apparent size of the super-genome, how the reservoir looks from the incidence of repeated gains.
Within this model.
There isn't much of a model here, actually. Oh, this is, yes, within the maximum likelihood model of genome evolution reconstruction. So basically, some conclusions from this. The genome dynamics are extremely variable, but typically are rapid. Rapid means rapid. There are thousands of genomic events per one nucleotide substitution per nucleotide, and many even per one nucleotide substitution in the entire genome, several at least. On average, loss has a higher rate, is more common than gain, and dramatically more common than expansion or reduction of families. Horizontal gene transfer, in other words, from this, horizontal gene transfer along with gene loss shapes the genomes. All rates are correlated, so gene flux is the key determinant of genome evolution. Typically, the supergenome is about 10 times the genome, but some supergenomes seem to be vast. But I think there are also general implications of this rapid genome dynamics. And to me, actually I must confess that I added this slide during this workshop. It was not there in the talk that I prepared, but I was induced to do so, compelled to do so, by the repeated discussions of Darwinian evolution here, Darwinian model of evolution. So of course, Darwinian concept is not one thing, it's quite a few, it's several important statements. But what we see here, that evolution does not proceed by infinitesimally small changes. And here is a rather famous quote from Darwin. Not the most famous, but rather famous quote from the original species. If it could be demonstrated that any complex organ existed which could not possibly have been formed by numerous successive slight modifications, my theory would absolutely break down. So Charles Darwin, parenthetically, was a great barbarian. He already knew that the important thing about the theory is falsifiability. So, this tenet, I think, is effectively falsified. It is not that, okay, you don't think so, Steve?
Well, it says complex organ. It doesn't say the basis of heritability. There's a distinction between genomes and complex organs.
All right, okay, take it. There is a distinction. There is a. Well, okay, you know. All right. I think a bacterium and an archaean qualifies as a complex organ.
But Darwin didn't necessarily think that way.
No, he simply did not think in that direction. Period. I think he would have been struck by the complexity of these organisms, especially their communities. So what we see here is that it's not that these are not the only types of events that occur. These not the most, by far, not the important events that occur. So I think that the issue is not so much whether complex adaptations could or could not have been formed by consecutive point mutations, but that they have not been, at least in this part of the world. And here I have finished this part of my talk, and already overdone the time, and there are two more important parts. So I will not, of course, go through them in the entirety, but I will simply say a few words about each. Basically, the phylogenetic forest of life tree, and that signals in the evolution of prokaryotes. So nowadays, we have transitioned, as I showed in one of my initial slides, in one of my first slides, from the study of the evolution of one standard, like 16 sRNA, to the study of evolution of huge ensembles of genes. They show different phylogeny. So the goal of this part of the study was to see whether or not there is some order in this forest of life. Whether or not there are some detectable patterns. I have to go through all of this. This was a detailed analysis of thousands of phylogenetic trees for prokaryotic genes, and the conclusions are that there is a common central evolutionary trend in this forest of life. So to put it, to encapsulate it in a tired aphorism, the tree of life is dead, long live the tree of life. So in the topologies of these trees, we can see substantial, significant order that can be approximated by the trees of what I call nuts, nearly universal trees. And these are the trees for highly conserved genes, including components of the translation system. So, by quantitatively, the tree of the 16 sRNA only shows a small part of evolution in the genomic universe. It was not a bad choice. It does represent some central trend.
This is on very large scale, is it? This is now.
This is on very large scale. On the scale of the entire, entire diversity of bacteria and archaea. There are many curious details, but unfortunately I cannot do that. We managed to develop a methodology to estimate the contribution of this trio-like signal and the net-like signal, the horizontal transfer. And roughly speaking, it's 1 to 2. One vertical signal, 1/3 vertical signal, 2/3 horizontal signal. So that's that. And the last part, which I will touch upon very briefly, the universal pacemaker of the genome evolution, general model to replace molecular clocks. Of course, we have talked already a little about molecular clock. Everyone knows what that is. Everyone, I think, in this room knows that it's over-dispersed, and therefore people now mostly deal with models of relaxed molecular clock. What we did was going further than that. And what we proposed is that the conservation of these distributions, okay, that we have seen, these are log normal distributions, and we have seen such in Steve's talk today. And these are the genomic distributions of the evolutionary rates of orthologous genes. They're strikingly conserved, strikingly similar from bacteria to mammals. I think that the conservation of the shape, of the amount of variation in these distributions might not be a trivial consequence of aggregation.
So what is this the distribution of? I guess.
Okay. These relative evolutionary rates of orthologous genes. And this is a probability density function, simply. So this is for bacterium, this is for humans, this is for a couple of fungi. So you take these rates over the entire set of orthologous genes in a pair of related genomes. And you plot the distribution.
The rates being the dn from the synonymous, ds.
You can do dn over ds. You can do dn. You can do ds. Only the width of the distribution will change. Specifically, these are amino acid substitutions. But you can do the same with ds. They're narrow, but you can do the same. So, given the over-dispersion of molecular clock, we sort of hypothesize that this is not a trivial consequence of molecular clock. And perhaps something that is more important than molecular clock and might replace it would be something that we denoted the universal pacemaker of genomic evolution. Namely, the model according to which it's the relative rate of gene evolution, not the absolute rate with respect to astronomical time, but the relative rate of genes with respect to each other that is conserved in the course of genomic evolution. And then we developed a framework to compare, to determine which models better fit phylogeny, the molecular clock model or the universal pacemaker model. In several experiments that we did across the diversity of life for bacteria and recently for eukaryotes, we show that the pacemaker model is substantially, I would say overwhelmingly, better fit to the topologies of these trees than the molecular quark model. So, again, from the genomic comparisons, variety of analysis, but again, from the genomic comparisons, we can see that apparently the rigid old molecular clock model can be replaced by a more general and more flexible, perhaps somewhat less unrealistic model of genome evolution. Basically, that's what I wanted to say. A tiny bit of philosophy before the research. First of all, this is not philosophy. I think this whole thing is extremely data-driven. The theory in this case, in my view, is extremely data-driven. We can transition to these perhaps more powerful models due to accumulation of massive genomic data and, I have to pat myself on the back, due to generation of appropriate data sets, such as those ATGCs, for instance, that allow us to test specific models. Well, and this philosophy, through the comparative study of evolutionary histories of numerous genomes and of genes they encounter, these new flexible models are starting to incorporate original regions but as special cases. Not unlike, I think, what is going on in physics in general. All right. So Sergei Sneer, who is here, the pacemaker man. Perry Puig is in your postdoc in my group. The tree man. Actually, the tree man and the ATGC man. Alex Wapkowski, the model man, and Yuri Wolfko, who is a senior member of the group contributing to all of this. Here we are in front of a building in Bethesda. Thank you very much.
Okay, maybe a couple of questions before the reception. Do you have any, or is everyone thinking about their free drinks? Yeah.
I was even struck by one of the first patterns you showed. There are these, these bacterial species or clusters which have these closed genomes and the ones which are open. So what explains that pattern? I mean, in the first place, before we go to
Well, the first approximation answer is I don't know. The second approximation answer is, of course, I can think of a few important factors. Basically, these factors are the lifestyle, that is, what is the diversity of the community in which the given organisms exists. What is the population dynamics? What is the population size of this organism? How much time it actually spends actively dividing rather than being dormant somewhere? Various ecological factors. And second important group of factors are actually more mechanistic. Do these organisms possess molecular machines such as those for DNA uptake? To what extent they are subject to bacteriophage attacks and so on and so forth? Do they encode so-called gene transfer agents, so on and so forth? Do they have if I may go back to something, do they have mechanisms for evolvability?
So the closed genomes, they have generally less mechanisms. That's your expectation to do uptake.
That is obviously my expectation, which is something that is that is to some extent already testable through the available diversity of genomes. But I must say that this homework has not been done, at least not by myself or by anyone I know.
So you distinguish two kinds of uptakes. I mean, uptakes within the group, and then uptakes from the super-genome, which is somehow...
No, I don't believe so. No, I do not. if I made that impression, I'm sorry. One could, you know, recalling Pavel's talk yesterday, one could revert to that kind of more complex model. But so far, I think we do not have the indications to go away from the now well-mixed model of the supergenome.
Okay, thanks very much.